Okay. Thanks for being here, everyone. My name is Michael Kramer. I'm from the Department of Medicine at UCSD, and I appreciate you all coming to listen. So the first question that I really wanted to address in this talk is why infer ontologies from data? This is kind of a new idea, and so what's the point of doing this? And in order to address this point, first I need to talk about the gene ontology, which is a project that we really have a love-hate relationship with. Now, the Gene Ontology project is an attempt to capture all that is known about how a cell functions in the form of a manually curated ontology. And so they represent this ontology as a hierarchical DAG. We can see a section of this ontology here, where the terms are the circles on this slide, and there are more specific terms representing more specific functions at the bottom, and more general terms at the top. And then for a number of different species, there are functions for genes annotated to each of these terms, which sort of gives you a map of what genes are acting in which processes in that cell, in that species rather. And so earlier I said we really have a love-hate relationship with this, and why did I say that? Well, we love the gene ontology, and we love it because it's useful. It's one of the most widely cited papers of all time. It's getting close to 10,000 citations at this point with no sign of slowing down. And clearly people are finding things to do with this. But still, we hate it. And why do I say we hate it? We hate it because it's frustrating. And it's frustrating because it's running up against the limits of manual curation. And this leads to several different issues. The first of these is coverage. If you look at the Go annotations for human, you'll see that 15% of human protein coding genes have no Go annotations at all. And that's frustrating for us because we know that every day people are generating high throughput data sets which tell you at least something about what these genes are doing. But when you look in Go, you don't see that. Second, this thing is massive. It's got tens of thousands of terms and term-term relationships. And in human alone, there are over 300,000 gene-to-term annotations. So it takes a lot of work for people to maintain this thing. They do the best they can, but of course you need multiple people working on this across many different fields, and that leads to issues of bias and consistency across the gene ontology. When you have multiple people with multiple ideas about what should be a term or what qualifies something for an annotation, it's really just kind of an intractable problem when you're trying to make something this huge using manual curation. And then finally, because it's so big, there's only really enough work to go around to make one of these per species. Now what I said before was that this gene ontology tries to capture everything that we know about how a cell functions. But of course there's more than one cell type in each species. And ideally, we'd like to capture how each of these different types of cells function. But there's just not enough manpower to do this manually. So how do we propose to address that? Well, we'll take the gene ontology and put it aside for a second. And instead, we'll try to infer a full hierarchical DAG, just like the gene ontology, directly from experimental network data. Now we know today there are many of these different types of interaction networks available in many different species, and there have been lots of papers that show that these networks contain functional information about pairs of genes or groups of genes and what they're doing. So we decided to put all of these together and infer an entire hierarchical DAG and see what it looks like. So in order to check what it looks like, we aligned it with the gene ontology, which gives us a one-to-one mapping between terms in our data-driven ontology and in the gene ontology. So with that mapping then, we can transfer information in both directions, which is why we have this arrow going both directions here. From the gene ontology to our data-driven ontology, we can transfer labels on terms that are aligned, which gives us some information about what each of these terms here represents. And then we can also take an overall global view and see just how much of the gene ontology we were able to reconstruct. And so in this first attempt, we showed that we were able to capture about 60% of the cellular component branch of the gene ontology. and to a lesser extent, we captured some of the biological process or the molecular function branch. But of course, our goal is not to exactly reconstruct the gene ontology. We already have the gene ontology, of course. So what we really want to do is find new things. So to do that, we look at the things in our data-driven ontology, which are robust, with the terms that appear despite perturbations in the data, but yet don't align to the Go. And so here's an example of one of those terms. It's Nexo 6164. It's got these 6 genes that you see here, which are supported by these different interactions. And when we looked in literature, despite the fact that this didn't align to anything in the yeast Go, we found this paper which implicates all 6 of these as the block 1 complex in yeast. It had previously been a known complex in humans, but only recently it was discovered in yeast and just hadn't been added to the gene ontology yet. And this is just one of a dozen examples that we found and then submitted to the gene ontology project, leading to updates in Go. So that gives us some confidence that things that we are finding in this data-driven ontology may be new and may be real. But of course, we don't want to just find the things that are already known in literature. We want to find things that nobody's found before. And so we're looking into that, and we have a few different tactics for going after these terms and giving us an idea of what they're up to. But I won't be talking much more about that today. If you want to look into some of these terms yourself, you can go to www.nextontology.org. Instead, what I'm going to be talking about today is where we go next. How do we improve these methods for inferring gene ontologies? So as you saw in this first attempt, we just performed an unsupervised learning process where we inferred this entire data-driven ontology. And it happened to capture much of what was known about the cellular components in yeast. But we want to be able to capture other types of ontologies too. One of the most interesting ones would be the biological process branch in Go, which we only captured about 25% of. We also want to be able to answer the question of what experimental network data is best for reconstructing an ontology or a specific type of ontology. And so we're thinking about these problems, and I realized that all of these things become easier to think about if we can factor our sort of ill-defined, take these and turn it into this problem, into 2 separate, more well-defined problems. And so that's what we're doing here. We take 2 different questions. One is, how do we take experimental network data and turn it into an integrated network? And second is, how do we take that integrated network and then turn it into an ontology? And that immediately leads to a question of what do we want this integrated network to look like? And I'd say that there are 2 main properties that we're really looking for here. We want it to encode the rich hierarchical information that is in an ontology. And that's sort of an open question. Is it even possible to encode a full ontology simply in a flat network? And second, we want this integrated network to be learnable from these experiments. So first, I'm going to address point 1. Can we encode a full ontology in an integrated network? And to do that, I'm going to try to perform this task, which is take the gene ontology itself, turn it into a network, and then turn that flat network back into the gene ontology. And so, of course, step 1 here is to turn the gene ontology into a network. And to do that, we'll use this Resnick Semantic Similarity score, which gives us a quantitative score between each pair of genes in the gene ontology. It's kind of represented here where the weights are higher for 2 genes that are contained in a very small term, very specific term with few other genes. And there'll be no edge or a weight of 0 between genes that are connected only through the root of this ontology. Then once we have that, I'm going to show you the Clixo algorithm, which can take this flat semantic similarity network and turn it back into a full, rich, hierarchical gene ontology. But in order to convince you that this is a non-trivial problem, first I'm going to show you what happens when we use other hierarchical clustering or other methods that can take a quantitative network and turn it into a hierarchy. So I'm going to show you the Nexo algorithm, which is what we used in our first attempt I talked about before. I'm going to show you several hierarchical clustering methods, and I'm also going to show you the local fitness algorithm, which can create a full DAG from a quantitative network. So I'll take each of these algorithms, I'll run it on the semantic similarity network generated from the gene ontology to get a reconstructed ontology. Then I'll align it to the gene ontology and calculate a precision and recall, where the precision represents the percentage of terms in this reconstructed ontology which actually capture something, a known term in the gene ontology. And the recall represents the percentage of the gene ontology that is being reconstructed by that algorithm. And what you can see here is that none of these algorithms come even close to reconstructing the full gene ontology from this network, which we derived exactly from the gene ontology. It's not a problem that we found any algorithm which could solve. And so we made the Clixo algorithm, which solves this problem nearly exactly. So how does the Clixo algorithm work? In order to show you, first I'll replace the gene ontology with a little toy ontology that we can all keep in our heads at once. The circles here represent genes, and the squares represent terms in this ontology, or functions in a cell. And when we take these, we'll take this toy ontology, calculate this Resnick score, which can be represented either as a network or as a matrix with scores between every pair of genes. And then now I'm going to show you Clixo. So Clixo is really only has a few main steps. It takes as input this similarity score matrix M, and it's going to output an ontology just like this. To initialize, we create a stack of sorted unique scores in M. So for each unique score in M, there's one entry in this stack. We have an empty undirected graph of all the genes, and we'll have an empty ontology O. And then there's really only 3 steps in the basic Clixo algorithm. First, it's iterative, so we're going to be going through each score in S one by one. Second, for each score, we find all pairs in M that are equal to that score and add an edge to the graph G, which is between the 2 genes. Then we find all the maximal cliques in that graph and add them as terms to O. And this will all be more clear, I think, once we walk through this toy example. So this is all the information on the last slide, but now I've got space to show you Clixo in action down here. We've got this stack S, we've got this empty graph G, and this empty ontology O, which we'll be adding terms to. So at the first step, we take the top of the stack, which is 1.39, and we add all the edges in M, which have a value of 1.39. That's these 3 edges here, each of which makes its own maximal clique, which you can see corresponds to known cliques, or known terms rather, in the original ontology. Next, we'll step through and get to 0.98, where we add edges between A, B, and C, creating this term 3 here. And this shows a couple of important features of this Clixo algorithm. One is that it can reuse genes. That's important because we know that in biology, genes are reused across different functions, and we wouldn't want them to be constrained to only one place. Second, it doesn't enforce any particular kind of splits at each level. By that I mean many algorithms enforce a binary tree only, but now Clixo is capable of doing a 3-way or even more way split if the data support it. Now just stepping through quickly, you can see the hierarchy starts to build here. Once we add the edges at 0.69, we create this new maximal clique between E, F, G, H, which combines the previous terms 5 and 6. And next step, we start combining multiples of the terms, building an even richer hierarchy. And finally, once all the edges are added to the graph, we get to the root. And you can see we're perfectly reconstructing this ontology down here. So of course, I kind of gave away the punchline on this slide before, But when we run the full Go biological process through the Clixo via semantic similarity scores first, Clixo is able to reconstruct the entire rich hierarchy in Go simply from this network with near 100% precision and recall, over 99%. So just to quickly recap why that was important and why we went through that test at all, what we've shown you is that the gene ontology can be transformed into a semantic similarity network using this Resnick score. And then we've shown you the Clixo algorithm, which can perfectly or near perfectly reconstruct the gene ontology from this flat network, illustrating that it is possible to encode this kind of rich hierarchical DAG simply in a network. Now I'll address point 2 that we want with our network, which is we want it to be learnable from experimental data. And that's the point of doing this in the first place, is to try to learn the ontology directly from this data. So in order to show this, we'll look at each of the few of these omics data individually first and see if they are consistent with a semantic similarity network. We'll look at 3 different types of omics data here. We have genetic interaction profile correlations. We have gene expression profile correlations, where we've calculated gene expression across correlation across all the microarrays in the Stanford microarray database. And then we have the YeastNet, which is integrated omics data from several different lines of evidence to give us a functional similarity score between genes. And you can see here that each of these types of data, represented on the x-axis, correlate in a noticeable way with the Resnick semantic similarity scores, which we calculated from biological process Go. And that gives us hope that it is possible to learn something like a semantic similarity network directly from these omics data. So now, of course, though, we want to run Clixo on these algorithms. But first, we need to make it robust to both noise and missing data, since this pseudocode you saw before is quite sensitive to both of these. There's really only 2 simple changes we had to make to do this. One is here in step 2. We had an alpha parameter, which has the effect of turning that hard equals into a soft approximately equals. When we do that and model noise on the semantic similarity network as just a Gaussian noise added on top, that gives us Clixo, which is very stable to in both precision and recall to various levels of noise. We also want to model missing data, which we do as setting values in the semantic similarity network to 0, and changing this third line here to nearly maximal cliques rather than maximal cliques. And that allows this Clixo algorithm to be very robust to high levels of missing data, 50, 60, 70% of edges removed, and we're still inferring something quite similar to the original ontology. So now that we've got a Clixo that's robust, we want to run it on the data. So first I'll show you genetic interactions. We saw before it's correlated with this Resnick semantic similarity. And when we run Clixo, represented as these colored diamonds here, it outperforms all the other algorithms that we tested. And a couple of things to note. One is that this alpha parameter acts as a trade-off between precision and recall. And then at each alpha setting, Clixo outputs the terms in order of confidence, so we have kind of a mini precision recall for each of these levels as well. Now you may notice that the recall overall is quite low. This is sort of to be expected. We're only using 1 line of evidence to try to reconstruct the full Go, and we wouldn't expect genetic interactions to be perfectly consistent with the Go. But still, Clixo is able to come closest. And we know that if genetic interaction profile correlation were perfectly capturing the same thing as the Go, the Clixo algorithm would be able to show that. Next, we look at the gene expression data on its own. And again, Clixo in the diamonds is outperforming the other algorithms. But of course, we don't want to just look at single sets of omics data. We want to look at bunches of omics data together. And so luckily there have been other people that have started thinking about this problem already. And one of these attempts to do this was the yeast net, which I mentioned before, where they take 9 different lines of omics evidence and create a functional network with this similarity score between pairs of genes. And so we saw before, this is correlated with the Resnick semantic similarity. And when we run Clixo and other algorithms, again, Clixo is consistently the best performer across wide ranges of the alpha parameter. So now that we've got this, there's several ways we want to go in the future. One of the main things we're interested in is how best do we combine all of these data into the semantic similarity network. I showed you one attempt done by another group, but we think we can improve on this. One thing that this will allow us to do is try to perhaps train to capture a specific type of ontology. And to do this, we could use the Resnick scores calculated from, say, the Go biological process and try to learn what relationships in the experimental network data support the information in Go. Then when we run it through the Clixo, we can find not only some of the things that are already known, but new things that are supported just as strongly in the network data, but have not yet been discovered either by biologies in general or at least added to the Go. Another area that we're really excited about is being able to take this entire pipeline and run it on a specific type of data from a specific condition. So for example, we can build an entire data-driven ontology for a healthy cell using only data from healthy cells and get this data-driven ontology which represents a full hierarchy of functions that that cell is performing. Then we can do the same thing on data from a diseased cell, align these two ontologies, and we can see how the cellular functions are changing, how the machinery has changed in disease. Of course, you can do this for many different types of comparisons. You control or some condition on cells. You can do this for different types of cells. And this is really an area that we're interested in exploring more in the future. So I just want to acknowledge my lab with Trey Eidecker and my co-authors Janusz Detkowski and Mike Liu, and also Vineet Bafna in the CS department at UCSD who helped a lot with this paper. And thanks to all the funding from my graduate education and also travel here today.
Hi.
Hi.
Fortunately for you, I'm one of the PIs on the Go. So,
Nice.
but I think this is very interesting work, and I do look forward to talking with you about it more. I have a couple concerns and a couple statements of misrepresentation of the gene ontology.
Okay.
So I'll do them to clarify some things about Go first, perhaps.
Okay.
One thing I want to say is you made a so of the 5 million annotations in Go, some very small percentages of those are manual. And yet what I'm hearing you're saying is that you're only using the manual annotations against the Go structure. So it's not the Go structure, it's the annotations that are informing your approach. Is that true?
Actually, in this work, we used both types of annotations.
So therefore, of those many millions of annotations, most of them are not manual. So your statements about the limitations of manual curation don't hold, because most of the annotations come from looking at the structure of the proteins and mapping to their domain structures through interproto-Go. So those are pretty strong annotations, and there's other annotations as well. So putting that aside, because that was your frustration with Go, and I just wanted to clarify that. And the frustration I heard was against the 15% of genes that human genes that have no annotations. And I just want to confirm for you and for others that 10 to 15%, maybe even more, of genes in any genome, even the most studied genome, we do not know what they do, period, the end. And we actually have domain determinations for those genes, and we don't know what those domains do. So we have a lot of knowledge. So I just wanted to make those couple of points, and but what I wanted to ask you primarily is how, I think this has a lot of promise for new areas of investigation. How are you thinking about the fact that all the data sets that you're using to build these systems have a tremendous number of false positives in them? So very rough data. It's preliminary data. It can lead to hypothesis generation, and that's maybe where you're going, but I'm concerned about that aspect of the data.
Yeah, so there definitely are false positives in the data, and we discuss a little bit in the paper about how we deal with them. One of the things that's nice about this algorithm is that it really relies on several different edges before it infers a term. And so the chances that false positives will align in a way such that there's a clique between 10 different genes, for example, I think is maybe lower, except for maybe in some certain cases. So yeah, that's definitely an area we're thinking about and looking into. I don't know that we have that solved yet, but it's on our radar for sure.
Yeah.
Before we have the next question, I want to interrupt. I believe that the next talk is not happening. That I'm going over to Phil. Is that correct? Or is she here? Okay. In that case, we have plenty of time for questions. I just and I want to make sure that I wasn't cutting off something here. So please continue.
Yeah, thanks. Pretty cool algorithm. This might be a dumb question, but I'm not entirely sure about the use case. I said, can you hear me now?
Yeah.
It's a cool algorithm. I'm just not entirely sure about the use case, and I hope this isn't a dumb question. But I mean, if your main use case is to sort of reconstruct an ontology from functional data, then the nodes of that ontology don't have labels, do they? So how do you know what any one node means?
Yeah, so there are a couple of ways that we try to get at that. One of the ways is that we use the information already in the gene ontology. So when we do this alignment from our ontology to the gene ontology, it gives us a one-to-one alignment between the nodes. And so we can just take the label from the gene ontology term and stick it on ours. And so that then when you look at the overall data-driven ontology, you kind of get a sense of what section of the cell you're looking at by looking at terms around it which have labels already. You can also perform kind of like a Go enrichment analysis, but with our ontology, which we've done as well. For example, in yeast, we looked at this data set that has several different cellular stresses and then tells you what genes are necessary under those conditions. So we can start labeling terms in our ontology as the genes in this term are necessary to deal with this type of cellular stress. But yeah, that's definitely an issue we're thinking about a lot.
Hi, hey, great stuff and interesting talk. And it's great to have the Go experts here. I think we recognize this is all really hard to try to understand biology. But we certainly have seen that we use Go and the tremendous amount of effort in the Go terms as the reference standard. And I think we can recognize and need to recognize that there are a lot of challenges in there. And, I just happened to look one up while you're talking, a really important immuno-oncology target, BAF3 TNF receptor superfamily 17, is annotated in Go as cellular proliferation. Well, certainly that's true, but it doesn't help you in figuring out the biology you're looking for. So what really is exciting about this is the ability for you to bootstrap and extend beyond that. And have you explored that further to get data to sort of expand on Go or even change Go and sort of refine it in a biological direction?
Yeah, definitely. So I think I touched on that for a second, but one of the things we're interested in doing is kind of feeding Go in as an input to this whole process. And that kind of allows us to, expand, do what you were talking about, expand on Go and use it as a scaffold to build upon. So that's one direction that we're taking this in. Yeah.
Hi, my name is Tudor. Very nice talk. One aspect I would be a bit concerned about is whenever I hear the idea of reconstructing ontologies, and especially with respect to Go, which is a bit more special than an ontology, is the fact that you don't have one single type of relationship in it.
Yeah.
So to me, it seems like, you're basically first, I won't pick on Resnick, which might not be the best sort of, score to use, but you're losing information when you're doing that transformation because you lose basically the type of relationship. And you cannot reconstruct that.
Yeah, so a
So
couple.
how does the evaluation then work, basically,
Yeah.
when you reconstruct?
So there are a couple, you made a couple points, which I'll talk about. One was this Resnick score. I didn't get into it today, but the algorithm's quite robust to using different types of scores. I just kind of showed the simplest one here. But yeah, your point about the relations is well taken. We have a couple thoughts on that. We've thought about that quite a bit as well. One point is just that in the average use case for Go, people kind of just use the terms anyway and don't use much of the relations. And I know that for a full ontology with computability, it's nice to have those relations. And so eventually we'd like to be able to try to infer those as well. But we think that there's a lot of usefulness that comes out of just this structure. And yeah, as far as inferring the types of relations, we have looked at, say, what types of network data would support different sorts of relations, and there are some things there, but that's sort of future work. We haven't solved the issue yet.
Thank you for the nice talk. My question is about the annotations with the experimental evidence. I'm sorry, the computational evidence rather. So did you consider the distinction between those two when you were measuring your accuracy of your Clixo algorithm? When you were reconstructing and measuring the accuracy of how it did in terms of reconstructing the annotations, right? Mm-hmm. Did you consider two different sets of annotations where some of them had experimental annotation and the others had computational? Because those computational annotations initially came from those types of data.
Yeah.
I'm
yeah,
sorry
no,
if this was mentioned already, but I just need a
No. Yeah, no, we didn't look at those separately yet, but that's a good idea. We should check on that.
Okay. All right. Thanks.
Actually, now I'm thinking, I think that was a supplement in our original, in the first paper from a couple years ago.
Okay.
But I have to go back and look.
All right. Thanks.